Thank you, Simonator. All right, let me bring you over here. Here we go. Okay, this is my condensed versions of notes for DNA to biotechnology. It's still a lot of slides. You should look at this in Google Classroom. This is meant to help you review for your test coming up on DNA and evolution. You should go to your notes. You should check those out. You should check out the Khan Academy readings I posted. You should check out the Bozeman Science videos I posted. And you should go back to your homeworks. The homeworks are good. Don't sleep on those on from the iPad book. All right. So it started out with trying to figure out what is the molecule that provides for hereditary information. So first experiment that we need to know, Frederick Griffith in 1928. All right, and just know the general outline. You don't need to memorize every little detail. All right, you look at a study guide for kind of some examples of it. So he studied these bacteria that cause pneumonia. And what he did was he mixed this bad strain, the pathogenic, that means it's harmful. This was the smooth one, and he gave it to a mouse and it died. Then he gave a different mouse, non-pathogenic, which was rough, and non-pathogenic means not harmful. He gave it to the mouse and the mouse lived. The mouse was okay. He then gave him this heat-killed pathogenic bacteria, so the smooth bacteria that's been heat-killed, heated up really high and made dead, and the mouse lived. Okay, so if we can heat up the bacteria, the mouse will live. So if we can heat up all this pneumonia bacteria, we'll live. Well, then he mixed the heated up pneumonia bacteria, the pathogenic, with the non-pathogenic or the rough one, and it ended up transforming the non-pathogenic one into something that can kill the mouse. So this dead bacteria that was heated up and dead transformed the rough bacteria or the one that's non-harmful into something deadly. So what causes bacteria to change or transform like that? Well, Avery, MacLeod, and McCarty tried to figure it out. And they used some enzymes to destroy proteins in a cell or DNA in a cell and see which ones were still transformed. Protease destroys proteins, DNA destroys DNA. So look, if they destroyed the DNA, the mouse lived. So that means that if they destroyed the DNA in the R1 and the mouse is okay. But if they only destroyed the proteins in the R1, the mouse still died. So you got to think backwards. This is a little tough. But if we destroy the proteins and the mouse dies, then proteins cannot be the transforming factor. But if we destroy DNA and it lives, so it had an opposite effect of Griffith's experiment, it can show backwards that DNA is the transforming factor. So Hershey and Chase kind of finalized this, and they confirmed it. So they did this famous blender experiment. Oops, I thought I had another page. They grew some bacteria, they grew some viruses in both sulfur and phosphorus, and in different weights of them. And what they did was they tried to see which ones are injected into the cell. Sulfur is only in proteins, it's in amino acids. And phosphorus is not in proteins and only in DNA, the sugar phosphate backbone. So if the phosphorus went into the bacteria, then it was DNA that was the transforming factor. If the sulfur went into the bacteria, then we know that it was the proteins that were the transforming factor. So they did it. They shook them. Here's the actual blender. You can go back to my notes from Classroom, and they confirmed that DNA is the transforming factor. All right, there's another person named Edwin Shargoff, and he showed that the amount of A bases, adenine, is always equal to the amount of T bases in an organism. Here's thymine and guanine and cytosine are paired up. This helped to kind of lay the framework for Watson and Crick and Rosalind Franklin. So that happened. I gave you my whole story about Watson and Crick and Rosalind Franklin. I'm not going to go through that again. But from those experiments, and then from the deduction work of Watson and Crick, and from the X-rays and the great work from Rosalind Franklin, we get deoxyribonucleic acid, or DNA. And it's up to you to figure out who or what gets credit for it. I think you probably felt my feelings in class. All right, so here's DNA. DNA's makes up chromosomes. You get 23 from mom, 23 from dad. You can see why genetics, DNA, proteins, meiosis, evolution is all connected, and it's beautiful and wonderful. A's always pair with T's and G's pair with C's. My ninth grade teacher taught me ashtray, garbage can. It just stuck with me. So whatever mnemonic works for you. Here it is. It's located in the nucleus. It coils up to make chromosomes. It's the blueprint of life. Okay, you should ask yourself, do you know what a nucleotide is? Pause the video. All right. Hopefully you know the nucleotide is made up of a deoxyribose sugar right here, and it's made up of a phosphate group and a nitrogenous base. This is one nucleotide. We put them together in dehydration synthesis reactions to add on to the DNA molecule. It's going to be with an enzyme called DNA polymerase. You can see that the A's and G's are bigger and the T's and C's are smaller. So A's had to pair with T's and G's with C's to make the helix have the right shape. These, we number the carbons on DNA, and you're going to have 5' and 3' ends. And that's just going to give, what it means is that DNA has a direction. That'll be on the next slide. And lastly, you don't need to notice, but the phosphates are negatively charged. So that's going to give DNA a negative charge overall, which helps us to do electrophoresis and some other cool things. All right, here we go. 5' to 3', 5' to 3'. They run in opposite directions. I will not test you on any 5' to 3' things, but you should know about them if you take some advanced biology. So the strands are anti-parallel. They run in opposite directions. They're held together by hydrogen bonds, just like water molecules are held together. So let's talk real quick about how they copy themselves, how it replicates during that S phase of the cell cycle. Well, first of all, it gets unzipped, and that unzipping molecule is called helicase. It's an enzyme. It ends in ASE, so we know it's a protein. Then enzyme called DNA polymerase is going to attach to it and start adding on the new nucleotides. There's other enzymes also involved, topoisomerase, gyrase, etc., but you don't need to know this for this course. But they do some cool stuff like hold the DNA open or release the tension, etc. So it's also checking it as it's adding on these new nucleotides, and eventually you get 2 identical copies. There's a famous experiment we went over in class called the Mendelssohn-Stahl experiment. The Mendelssohn-Stahl experiment shows that DNA is copied semi-conservatively. So that means that each strand is going to have 1/2 new and 1/2 old. So it keeps the same information. So this is pretty cool. And this is why DNA is able to provide your heritable information. There are leading and lagging strands. I showed you some Okazaki fragments being made in class. Those you won't need to know in a test, but the lagging strand is going to need special enzymes because it can only be added to the 3' end of a molecule. In eukaryotic cells, it happens at hundreds of spots. So those are called replication forks. Whereas in a bacteria, it just moves in both directions until the chromosomes completely copy. Here you can see the prokaryotic one happening. And here you can see these replication bubbles in a eukaryotic. That way it wouldn't take days to copy our DNA, but we can do it quickly. Okay, let's make some proteins. Here's our DNA. We're going to make RNA. We're going to remove the introns, and then it's going to go to the ribosome and make a protein. You all did really well on this in class, and you picked it up super fast with the codon charts. You need to know a little bit about RNA. You should know that it's single-stranded. It's got the sugar ribose, so it has 1 more oxygen, and it uses the base uracil instead of thymine. You can remember that because RNA, students, loves you. Very nice. Oh, I'm trying to make a heart. There it is. RNA loves you. Okay, see if you can come up with the RNA for this DNA. Pause the video. Whew, it didn't come up. It would have been UAU-CGC. All right, here's mRNA. This is the messenger. All of our proteins are going to start with AUG when we read that chart. All right, that's the start codon. And then each triplet, it's a codon. It's read in sets of 3, is going to code for 1 amino acid. Here's what a ribosome looks like. It's made up of RNA and proteins as well. And so it's called ribosomal RNA. And here's what tRNA looks like. So this is one of the few instances when RNA is not single-stranded. It's being held together by bonds, and it's making what looks like a T or a cross. At the very bottom is going to be what's called the anticodon. The anticodon's going to match up with the codon to provide for the genetic code, right? So if this is CCG, the anticodon has to be GGC. That'll bring the correct amino acid in here, which is alanine. All right, so how does it make the protein? DNA to RNA is called transcription. RNA to protein is called translation. Proteins are the building blocks of everything pretty much we do. So you need to know transcription and translation. Now, I don't have it in this video, but you should be able to go and given DNA, you should be able to make RNA and you should be able to make proteins. Most students will ace that part of the test. So just look over the worksheet we did with that. All right. What happens if UV light or something hits our DNA and messes it up? Well, we can have mutations. So we went over point mutations. You can have a substitution. Here, the T got changed to a C. You can insert or delete a nucleotide, which is going to cause a frame shift mutation. And these can cause changes, really bad changes to the protein. You can also have chromosomal mutations as well. Here's a substitution. Here's an insertion. Here's a deletion. So I think in class you liked it when I said the fat cat ate the hat. So you could change it to the fat rat ate the hat. That'd be substitution. Or insertion, the fat cat, but then we might add another C after the C and cat. So it'd be the fat cat tat ate the hat that. So you can't really get the protein out of it. And same for a deletion. What are the effects of it? So we went over this quickly in class. You should pause the video and ask yourself, do you know what the effects of a mutation can be? It can be silent, as in it doesn't change it. So here's the DNA ATG. Here's the RNA UAC. It's coding for the amino acid tyrosine. Here we have a mutation. We go from ATG to ATA, becomes UAU. It still codes for tyrosine. So this is a silent mutation. All right. It could be a nonsense. Nonsense means it stops it. Bam. So we could make an incomplete protein. It changed it from UAC to UAG, which codes for a stop codon. So just like you might have said for prom, stop dancing, UAG says stop making the protein. Or it could have been a missense. So it's going to change the amino acid. Here that G, the A was mutated to a G, became CAC, and it becomes histidine. So it's the wrong amino acid. If it becomes a non-polar amino acid and one was polar, that could be really bad. Here you can see the chromosomes being flipped around. We didn't get to it, but I was going to have you guys look up different diseases that are associated with all of these, but that's something to check out on your own time. Okay, it can be spontaneous. It can be an error during DNA copying, so it can't be perfect. Think about how many DNA bases you had. You saw watch that TED Talk with the books full of DNA. It was insane, right? Or it can be induced, and this is where things like smoking or UV light can do that. So you should avoid X-rays, smoking, too much UVs. Okay, we talked a little bit about gene regulation. We talked about eukaryotic gene regulation. That'd be in U, and how you have RNA interference and you have the dicer enzymes and stuff, but you don't need to know that. Okay, so that was an extra. What you do need to know is the lac operon. Lac operon is like the lights in this room. Like I'm about to leave in a couple minutes to go pick up my kids. I'm going to turn off the lights because there's no more learning happening in here today. It's happening at your house. Here, if there's no lactose present, this is turned off. So a regulator gene upstream is going to make a repressor protein. Notice that a repressor protein can fit in beautifully right here, and it stops the transcription of these genes. Here's where RNA polymerase would bind on, and it would transcribe these genes. But if the repressor's here, no go. Now let's say that students are at school. I'm going to turn on the lights. Well, let's say that lactose comes into the cell. The lactose is going to come here and bind with the repressor protein, and it's going to be removed. When it's removed, then transcription takes place. So we're only turning this gene on, we're only inducing it to be on when it's needed. This is very important for a cell because it lets the cell save energy. You can remember this because of PROG. So some of you all watched the Amoeba Sisters or the Bozeman video on this. It's a good thing to review. So you have the promoter, you have the operator, the on-off switch, the repressor, and then the genes. You do not need to know eukaryotic gene regulation, so you can wipe that out. But this is some pretty cool stuff. All right, last but not least, biotechnology. So now we know about DNA. We know about mutations to it. We know about how it's regulated. Sorry about that. So now we can edit it. We can work with it. So there's these crazy things called CRISPR that we'll get into at the end of the year. For us, what I want you to know is that we can cut up DNA with restriction enzymes. It's going to cut whenever it sees the sequence it's designed to cut. These are enzymes that we get from bacteria. The bacteria use them to cut out viral DNA that infects them. We can use it to cut a plasmid or a bacterial, a small piece of DNA in a bacteria. We can then cut your DNA and we can insert it into the plasmid. So we can get this to, we can grow human insulin by taking this plasmid from bacteria, cutting it, cutting the human gene for insulin production out, putting it into this plasmid, putting it back into a bacterium, and then it can reproduce. Bacterium can pass on these plasmids to each other. They can engage in what's called lateral gene transfer, which is pretty cool. So that's horizontal transfer. So they pass them on, and then these proteins get read. All right, so we use that to, we can insert genes for pest resistance into plants, cleaning up toxic waste, human growth hormone. We use this for a lot of really cool stuff. Okay, we did this in class. So gel electrophoresis. You should be able to tell which one of these matches up to the blood stain here. So which one matches up to the blood stain at the crime scene. You can look up the OJ trial and see this happen too. Here we can see that 3 matches the blood stain. So we get this by chopping up your DNA with those restriction enzymes. Your DNA is going to be different than mine. So if we run them through the gel, it's going to go towards a positive electrode. Your band sequence will be different than my band sequence because of where the restriction enzymes cut our DNA. All right, so that's gel electrophoresis. We just talked real quickly about that. You can watch it with this animated GIF right here. And then PCR, this will just be a bonus on the test if it's on there at all. But we can make many copies of DNA. So we can heat it up. When we heat it up, the hydrogen bonds break. We use an enzyme called Taq polymerase to add them on there. That's just the bacterium we got it from. It's just like DNA polymerase. It's going to add the new nucleotides. We then lower it. We lower it so that can happen. Then we heat it up again. And we can make over 2 to 4 hours, we can go from a single copy of DNA to billions. So this is why if you leave one hair behind at a crime scene, you can get caught. All right, students, that was it. That was super quick. Sorry, I got to pick up my kids, but I hope it was good for you. And good luck studying for your test. See me for Sunday's review session if you need help. Thank you.